Hello, my name is Phillip, and I'll be presenting my poster titled Non-Invasive Targeting and Stimulation of Individual Brain Networks. So brain networks can be identified by measuring brain function at rest, also known as resting state functional correlations, or RSFC. Brain network organization differs across the lifespan and in disease states, and these differences, importantly, are related to differences in cognitive ability. There's also substantial individual variability in brain network organization, even in homogeneous groups like healthy young adults. There's been an increased interest in leveraging non-invasive brain stimulation techniques as a potential method of treating various psychiatric disorders, particularly depression. However, research in the utility of such therapeutics has yielded uneven results, and this might potentially be due to the heterogeneous nature of the stimulation targets that they're employing. So in our study, a total of 17 healthy young adults completed all study procedures. Participants would come into the lab for 7 consecutive days. On the first day, there would be an MRI scan session. On days 2 through 6, participants would receive 20 minutes of non-invasive brain stimulation, specifically transcranial magnetic stimulation. On the 7th day, a follow-up MRI scan was completed 24 hours after the final TMS session. Participants would also return for a second week of sessions after about a 4 to 8 week break. Each of the weeks, participants received stimulation to either the left angular gyrus or the left middle frontal gyrus. Importantly, the left middle frontal gyrus is a typical target used in depression therapeutics. Each week, the opposing target was treated as a control site. So targets were selected on an individual basis. Each subject's MRI data were used to create individually specific functional brain areas, which were then assigned to different functional systems in order to construct our brain network. So it's important to note that individual targets can vary. Again, this left middle frontal gyrus, which is typically employed in depression therapeutics, it has quite a bit of variability. In these 3 participants here, they have a yellow frontoparietal brain region within this location, whereas this subject does not have the frontoparietal system available. For this subject, stimulating this area would potentially yield different results. So for this subject, the TMS coil would have to be placed in a different location. So it appears that stimulation-related changes in RSFC strength might be related to the baseline strength between 2 different regions. So here are the distributions of the angular gyrus and the middle frontal gyrus before and after stimulation. We can see that after stimulation, the distribution appears to be getting squeezed, where stronger positive and negative correlations are becoming weaker. But we don't see this when we look at the other control site. This can also be observed in an individual example subject. There's a very strong relationship between the baseline RSFC and the change in RSFC for both the angular gyrus when an angular is being stimulated and for the middle frontal gyrus when it's being stimulated. However, this relationship does not exist for the opposite control target. We can see this statistically here. Positive and negative connections are separated so that we can highlight the difference in the directionality. For positive connections, we can see that there is a decrease, significant decrease, in resting state functional correlations for the middle frontal gyrus, but only when the middle frontal gyrus is being stimulated. Similarly, for negative connections in the brain, the middle frontal gyrus connections are significantly reduced when the middle frontal gyrus is stimulated, and the angular gyrus connections are also reduced, but only when the angular gyrus is being stimulated. Similarly, the change in RSFC seems to be dependent upon system. If we look here on the right, we can see that within a specific system, for example, the default network, the RSFC values decrease significantly after stimulation, but this is dependent upon the stimulation target. So in the default network, there's only a significant decrease after the angular gyrus has been stimulated. And in the middle frontal gyrus, in the frontal parietal network, there's only a significant decrease after the middle frontal gyrus has been stimulated. So taken together, we can conclude that brain areas that have similar functional properties may actually be located in slightly different anatomical areas across people. And this would necessitate the use of individualized stimulation targets. So importantly, the impact of the stimulation-related change to a specific connection might be related to how strong that connection is at baseline. And changes in resting state functional correlations are also specific to the stimulation target and the functional brain system. So taken together, this suggests that there should be appropriate selection of a stimulation target, as this may be critical to the clinical outcomes when used to treat patients with potential depressive symptoms.